Hi, my name is Owen Dabkowski, and I am a master's student at the University of Otago in New Zealand. I'm super excited because I've been working with the Dabkowski project to create a new method to measure prey size from animal-borne camera footage. The Dabkowski project was co-founded by Thomas Mattern and Ursula Ellenberg to study the elusive fjordland-crested penguin, also known as Dabkowski. To do this, they created their own line of animal-borne cameras, which were small enough and lightweight enough to be deployed onto penguins. These cameras are super cool because they allow you to get a first-hand experience of what the penguins do during their foraging trip, including what they eat, what they interact with, and where in the landscape they are foraging. Currently, when studying diet, composition and contribution is determined by fecal matter and stomach contents. However, the new method we created to study diet composition will allow for the energetics of the foraging trip to be fully understood. This can help you determine why penguins target certain species over others, or why they may feed in certain areas. For this project, we took some penguin cameras and made a series of measurements of a known object at a known distance. The known distance was the distance between the penguin's beak and the camera lens when the cameras were deployed. Using these measurements, we created a correction factor. The correction factor is a pixel to millimeter measurement, which allows you to take a freeze frame from your footage just before the prey is eaten, and then measure how long or tall it is in pixels, which you can then convert to a millimeter measurement, which allows you to use it in an energetics calculation. This means that you can determine how much energy the penguin is gaining from each foraging trip versus how much it is expending, helping to provide insight into why they target certain prey or certain areas. One of the most interesting things that we found is that salinity did not have an effect on correction factor. This meant that while we predicted that you would need to have a different correction factor for each salinity, this wasn't the case. As a result, the correction factor is so much easier to use because you don't have to worry about pairing your camera footage with a salinity logger in order to work out which correction factor you need. Instead, all you need to do is work out whether the camera is in air or in water, and you need to take a beak measurement and a known distance measurement when you deploy it in the field in order to be used. The other interesting thing that we found is that distortion was different between air and water. So in water, the correction factor for the known object on the edge of the image was larger than in the center. This is known as barrel distortion. Whereas in air, it was the other way around. So the correction factor for the object was larger in the center than on the edge, which is known as pincushion distortion. This meant that we had 4 different correction factors. So we had 2 for air and 2 for water. One in the center of the field of view, and then one on the edge of the field of view. The correction factor is super cool because it allows you to measure something as small as a fish larvae, which is only a couple of millimeters long, all the way up to something that fills your entire field of view, like a tuna we saw in king penguin footage from South America. One cool thing about this paper is it provides a new way to study marine predators. It allows us to understand diet composition, but more importantly, it allows us to calculate energetics to determine what drives the decisions penguins make during foraging, which is something that has not been done in this way before. This new way of utilizing animal-borne camera footage can help us better appreciate the complexities of the marine world. It can be used to assess risks from human interactions and inform new policies which can help benefit the species, helping with conservation and management of these important animals.